Hi, my name is Rachel Sullivan, and I'm a fourth-year neuroscience student at UCSF. I'm really honored to be an ARC scholar, and I'm excited to share my project with you. At UCSF, I'm in Mason Carebeck's lab, and in the Carebeck lab, I'm trying to understand avoidance behavior, which is a characteristic feature of many anxiety disorders. We use a mouse model to study avoidance, which allows us to directly probe the functionality of relevant circuits. And our hope is that what we learn can one day be applied when developing treatment for humans with anxiety disorders. So what we have learned in our lab and in other labs is that the ventral hippocampus, and specifically the VCA1 subregion of the ventral hippocampus, shown here in green, is an important driver of avoidance behavior. And classically, we've used this maze shown here to probe the contribution of VCA1 to avoidance behavior. This maze is called the elevated plus maze. It has two shielded dark arms and two exposed brightly lit arms. These exposed arms are inherently aversive to the animal. They don't like being in bright exposed spaces. So when you let a mouse freely explore the elevated plus maze, it will tend to stay in the closed arms and avoid venturing into the open arms. And using this tool, we've learned two main things about VCA1 and avoidance. One is that the activity of VCA1 increases when animals enter those open arms. And two is that if you inhibit VCA1 when animals are in those open arms, they'll stay in those open arms for a lot longer and stop avoiding them as much. And so what these experiments have shown is that there's a clear role for VCA1 in orchestrating avoidance behavior. But the elevated plus maze has its limitations. This tool only measures innate avoidance. Mice are born averse to environments like the open arms, and there's a whole other class of avoidance behavior that isn't yet understood, and that's learned avoidance. So avoidance of cues that are inherently neutral, but become associated with an aversive outcome. And this is what I'm interested in studying. How does VCA1 orchestrate avoidance of learned cues that predict aversive outcomes? So to explore this question, I've set up a learned avoidance paradigm in our lab. And in this task, the animal's placed in a two-chamber box. A light flashes underneath one side of the box right before a shock is delivered to that same side of the box. And if the light flashes under the animal needs to move to the other side of the box in order to avoid being shocked. And we call that an active avoidance trial. The light flashes on the opposite side to the animal just needs to stay put in order to avoid being shocked. And we call that a passive avoidance trial. And after setting up this experiment in the lab, I wanted to see what happened if we inhibited VCA1. Do I see the same impaired avoidance response as I see in the elevated plus maze? And so I wanted to use something really strong to inhibit those VCA1 neurons. And so I used a tetanus toxin to permanently silence VCA1. And you can see that here, the tetanus toxin infected neurons are showed with the green fluorescence. And what I found here is that this inhibition produced an effect opposite to that observed in the elevated plus maze. So in this task, inhibition promoted avoidance behavior. You can see in this graph the average percent of active trials that animals appropriately avoided. And you can see that the tetanus animals shown in red learned to avoid active trials significantly faster than control animals. And this was also true in passive avoidance trials. And so now I'm trying to understand why VCA1 would have such different effects in the elevated plus maze versus the learned avoidance task that I'm using. And to try and investigate this, I'm doing calcium imaging experiments to understand the dynamics of VCA1 as cells engage in the learning task. So you can see here an example field of view where the neurons are brightly lit, and you can see on the right example traces from these neurons. And so that's what I'm doing right now, just processing this data to try to understand exactly what VCA1 is doing during learned avoidance. So that's what I've got for you today, and I'm excited to share more at the symposium, at my poster. I want to thank ARC so much for all their support and for making this research possible.